My name is Joe McAuliffe. Wrong direction. I work for IFF, but today I'm actually representing ASTM because I'm also the chair of one of ASTM's numerous technical committees, E62. I'll be talking about our efforts to develop standards for industrial biotechnology, synthetic biology, whatever you call our evolving field. And really one of the major take-homes of the message today is how important standardization is to this overall theme, how good standards can lead to good policy, not necessarily, but I think it's a prerequisite in many cases. Regulatory frameworks require good standards. An example, Richard talked about, if we have an earthquake, get under the desks. I'm wagering the building won't fall down because ASTM standards dictate how many materials should be tested and what they need to conform to. ASTM, which I will introduce in a second, this is an overview of the talk. ASTM was originally founded in 1898 to bring standards to steel because there was wide variation in the quality of steel. Was originally called the American Society for Testing and Materials, and now it just goes by ASTM International. It's a large organization, non-profit, volunteer-driven, quite different from ISO, which is another large standards development organization in how it operates, but with the same overall mission, to basically drive the expansion responsibly of many, many industries through the setting of standards. I'll explain a little bit more what I mean by standards and what forms they can take. Anyone can join ASTM. It's about $75 a year. You can participate at any level from a task group where you don't actually have to be a member. But if you are a member, then you can vote on standards. One aspect of the ASTM process is that any negative vote has to be considered. They can be overridden, but it's quite a process. But it's one of these well-defined processes that we think gives legitimacy to the process of setting standards. So here is ASTM Committee E62. We were founded in 2014 with the mission to bring standards to, at the time, industrial biotechnology. I'll talk a bit more about what we mean by that. And we aim to develop, various types of standards for nomenclature, definition of terms, best practices, guides, test methods, etc. You can see the faces here. Steve Evans is one of the members. He's here today. Reena Singh, who also is an exec VP with the American Fuels and Chemicals Coalition. And Pat Piccarello is our staff manager. So the mission here is to develop standards that promote the expansion of industries, plural, because as I'll explain in the next slide, industrial biotechnology is not an industry per se. It's a means to an end. It impacts many different industries. And we introduced synthetic biology recently, 2 or 3 years ago, into the name of our committee, because this is increasingly being used, and here's some terminology definitions, as what we used to call maybe genetic engineering, we're now calling synthetic biology. Synthetic biology began as really, in many ways, an engineering philosophy. Can you turn biology into these modular units so you can swap pieces in and out? Now it's kind of evolved to be maybe all of the above, a rebrand of genetic engineering. In contrast, industrial biotechnology is, again, we call it an industry, but it impacts many things: cleaning, food production, agriculture. Many things can be put into that overall bucket of industrial biotechnology. But together, it represents a very important component of the global chemical value chain and other industries beyond just chemicals. So terminology is really important. And I think it's very important that we, when we're explaining to the public and other stakeholders, we explain our terminology. I can't emphasize how important that is. So here's a provocation. Has standardization lagged in industrial biotechnology? I would say yes. There's many reasons. Humans didn't invent biotechnology. When people say, well, look at the rate of progress, it's not like we've seen in tech, we're reverse engineering an alien technology 3 billion years old, at least. I think we've done very well, actually. But in many ways, that really dominates how you come at standardization when you're dealing with natural systems that you're adapting to your purpose. And so in many ways you could argue development of standards has not kept pace with policy and regulation, and that's a problem. And so part of our mission is to try to remedy that to some extent. So another question is, where are standards needed most at this point in time? I mean, one of the focuses of this workshop, of course, is containment of organisms that will be engineered and released. That certainly is something that we saw, I guess, some time ago as a critical area for where standardization is needed. And so I'll talk more about that in a few slides. I apologize for the formatting. It's kind of gone a bit cattywampus, but anyway, standards come in many flavors. There's many bodies that standardize. ASTM is just one of them. They operate a little differently. I like to break things down into strategic versus tactical standards. Tactical might be test methods for a very specific material. Important, but not really something that would influence, for example, policymakers and other non-technical stakeholders. That's where a strategic standard, one that maybe makes people consider things in a different light. And so in many ways, E62 has focused more on the strategic. Also volunteer-driven, it's not our day job, there's only so much we can do. I'll give an example of that, a microbial classification system that we developed back in 2019 and we're currently revising. And as mentioned before, there's many different types of standards, often quite high level. They become more detailed over time. Others begin, for example, the standards developed by D20.96 for biodegradability and test methods to assess biodegradability. Very complex standards. So current standards and active work items for E62. So I'll talk a little bit more about the standard classification for industrial microorganisms. We do have a terminology. It consists of over 60 terms. Part of ASTM's business model as a non-profit is you have to pay for the standards. It's a modest amount. It's about $70 per standard, but we can't just release them to the public because that's not how ASTM earns its money. But anyway, I do encourage, if you're interested, I do have high-level overviews of these standards, which I'm quite happy to share with people. We're also working on standards that, again, are getting bogged down in detail. For example, how to determine the carbon intensity in regenerative agriculture, physical test methods in that case. This is one that I'll talk a little bit more about. We've gone around in a few circles here. This was Zach Abbott from Z-Biotics. How do we come up with best practices when you are going to engineer and create an organism you intend for environmental release? What are those guidelines? Zach puts it for the eager but naive player. What are the pitfalls? How do we guide them best? And another one that's moving slowly, but hopefully we'll get this one out there as well, which is how do especially investors judge the risk of being able to access biomass when they're thinking about building, for example, a plant that uses biomass as an input? So these are the current items we're working on. Let me talk a bit more about the standard classification that we developed and we're currently revising. The motivation for this was we did see that there would be increasingly more products on the market where it would trigger questions in the minds of all stakeholders, not the least of which consumers, in terms of what is this product, what does it do. We really wanted to move the debate beyond, is it GMO, yes or no? It's still a digital debate. We don't think it's helpful. And so this is the field, this is the classification as it stands. I'll describe in a few minutes some of the upgrades we're considering. But the first field is genotype class. And we've divided this into 4 different categories. Quickly, a class A is not phenotypically selected with intent by any means. Class B is there's been some phenotypic selection by any means. We're agnostic as to how, right? But within the observed genetic diversity at the species level, this is at a species level. A class C is where you fall outside observed genetic diversity and there's still been obviously some intent for phenotypic selection. And a class D would be an organism engineered outside of that observed diversity where it is making a chemical not known to occur in nature. So we tried to expand the categories that you would call genotype class. Field 2, we just borrowed from the NIH biosafety guidelines because we didn't want to be in the business of setting safety and risk. We are changing that. I'll tell you how. Field 3, mode and primary application. What is the mode of use? Is it contained kill? Is it to be sprayed on fields? Is it for human consumption? There's about 6 or 7 modes. And primary application, because sometimes things have a secondary application, like an ethnologen for making ethanol has a secondary application as animal feed. But currently, we just list the primary application. And then field 4, depth of DNA sequencing. We're looking at expanding that now to a more broad characterization and history of use in general. So this was balloted in 2019, active standard. Companies haven't been rushing to adopt it. Maybe they will. But as a means of provoking discussion, I think it has a lot of value. And hopefully companies will adopt it. This is how we're modifying it. We are going to leave field 1 as is. We are going to change field 2 to trait risk. This was Vince Seawalt's suggestion. Biosafety risk grouping per NIH is directed mostly to pathogenicity for humans. And also, are there available measures, right? It is not directed to environmental harm. Nearly all industrial microorganisms are BSL level 1 and risk grouping 1. So we wanted to change this. We're still wrestling to some extent exactly how to define trait risk. So negligible, low, medium, high. Any BSL-2 or higher is automatically a high trait risk. How do you place antibiotic resistance markers? A lot of human probiotics you can buy in the store have antibiotic resistant markers in them. So we're still wrestling with how to define this, but we think in the context of industrial microorganisms, this would be a better way to go. Similarly, we're changing field 4. We want to expand that characterization beyond just sequencing, so phenotypic characterization, omics, proteomics, metabolomics, but critically also use at scale. They tend to correlate. If you've got an organism like Bacillus subtilis that's used to make dozens of different products, it's been very well characterized. And so this would be 1 to 4. Something that's in the laboratory, never been deployed or even scaled to pilot would be a 1. An organism that's maybe used to make 2 or 3 or less products, 1 or 2, maybe use a 3. So how we define this, a lot of this is subjective, obviously. This system would be a manufacturer's declaration. However, the system only applies to products that have DNA in them. So there's some degree of opportunity for verification. And again, the overall questions that hopefully talking about a system like this generates, I think, is part of the value. So I can give people more detail if they're interested. So new standards under development, one of them I pointed out was, how do we generate a standard, which initially would be probably a high-level standard, that offers guidelines when you're trying to build or planning to build an organism that would be deliberately released into the environment. And so, drawing on this report that Richard and others talked about, which gave some policy recommendations, how do we bridge this gap to a standard? And that is something I would like to put to you all. I'm looking for suggestions, input. How do we do this? What would these guidelines look like? I mean, there's obvious ones. Don't be leaving antibiotic resistance markers in your strain. Not everyone knows that. Most people would. Other things like, don't be using an organism that has mobile gene elements in it. But we need a bit more nuance than that. But it would begin at a relatively high level. And so I am hoping that we can align our efforts because there are a number of different groups, not just this one here, Lynn Center, who are thinking about this. And there's a lot of parallel efforts right now in the area of standardization within biotech. And I think we need to align somewhat, and that's one of the things I'm hoping to get out of this meeting as well. Who else should we consult? So anyway, last few slides. This is the priority actions for our subcommittee, is to revise and reballot the standard I talked about, the microbial classification, to expand our efforts to have industry adopt this standard. right? That would be wonderful. Big companies like the one I work for are pretty conservative, move slowly. Smaller companies like Biotech, I think maybe would be more likely to adopt a standard like this. But whether this ever ends up on a consumer label, that would be great, but hard to predict whether that will happen. The carbon intensity standard, which is incredibly detailed, lots of formulae in there. Ramani Narayan is one of the drivers of that, Michigan. Hoping we can get this to ballot at least next year. Standardization moves very slowly, by the way, and it can be quite frustrating, but we think it's worth it in the long run. The supply chain risk standard, development of the standard I just talked about for guidelines for engineering organisms, and to grow our membership. So if you're interested, you can join at any level. Again, even just task group participation does not require membership, and the amount of time you put in is up to you. But there's all the way up to joining our executive committee. So how do you work with us? As I said, you could join as a member. You can participate in task groups if you wish at any amount of commitment. Consider joining as a subcommittee member. And please do consider working with us because, I think we share a lot of common goals here. So with that, I'll conclude. Thank you.